Good morning, everyone. We shall talk about the topic infectious diseases. This shall be taken in two classes. Now, today we'll take up part 1. Coming to introduction, microorganisms, namely bacteria, viruses, fungi, and parasites, are present everywhere in the soil, water, atmosphere, and on the body surfaces and are responsible for a large number of infectious diseases in human beings. Some microorganisms are distributed throughout the world while others are limited to certain geographic regions only. In general, tropical and developing countries are particularly affected by more infectious diseases than the developed countries. There are several examples in certain diseases which are not so common in the developed world, but they continue to present as a major health problem in the developing countries. Example: tuberculosis, leprosy, typhoid fever, cholera, measles, pertussis, malaria, etc. Vaccines have, however, been successful in controlling or eliminating some diseases all over the world. Example of smallpox, poliomyelitis, measles, pertussis, etc. Similarly, insecticides have also helped in controlling malaria to a great extent. Besides microorganisms, a modified infectious host protein present in the mammalian CNS has been identified called prion protein. They are responsible transmissible agents similar to the infectious particles, but they lack the nucleic acid. They are implicated in the etiology of spongiform encephalopathy, bovine spongiform encephalopathy, or Creutzfeldt-Jakob disease. Transmission of the infectious diseases require a chain of events and in a sequence of interrelationship between the disease producing properties of microorganisms and host defense capability against the invading organisms. Now we shall talk about the chain of transmission in the infectious diseases. Number 1, reservoir of the pathogen. Infections occur from the source of reservoir of the pathogen. It may be human being, animal, insect, or soil. Coming to the next, that is the route of infection. Infections are transmitted from the reservoir to the human being by various routes, usually from the breach in the mucosa or the skin, and at the portal of exit from the reservoir as well as the portal of entry from the susceptible host. Coming to the mode of transmission, the organism may be transmitted directly by physical contact or by fecal contamination, or indirectly by the fomites. Then coming to the susceptible host, the organism would colonize the host if the host was good immunity, but such a host can pass on infection to others. If the host is old, debilitated, malnourished, or immunosuppressed due to any etiology, he is susceptible to have any manifestations of the infection. The key of management of infection lies in the breaking or blocking the chain for transmission and the spread of infection. Now coming to the factors relating to the infectious agents. Number 1, that is the mode of entry. Microorganisms causing infectious disease may gain entry into the body by various routes like ingestion, that is the external route, inoculation, parenteral method, inhalation by respiration, perinatally by vertical transmission, by direct contact, that is contagious infection, and by contaminated water, soil, environment, or from animal host, that is the zoonotic infections. Then coming to the spread of infection, microorganisms after entering the body may spread further through the phagocytic cells, blood vessels, and lymphatics. Virulence of the organisms. Many species and strains of organisms may have varying virulence, like the 3 strains of cholera diphtheria, gravis, intermedius, and mitis produce the same diphtherial enterotoxin but in different amounts. Then coming to the production of toxins. Bacteria liberate toxins which have effects on the cell metabolism. They are liberated on lysis of the bacterial cell, while exotoxins are secreted by bacteria and have effects at distant sites too. Then coming to the product of the organisms. Some organisms produce enzymes that will help in spread of infections like hyaluronidase by Clostridium welchii, streptokinase by streptococci, etc. Now coming to the factors relating to the host. Number 1 is the physical barrier. A break in the continuity of the skin and mucous membranes allow the microorganisms to enter the body. Then comes the chemical barrier, where the mucous secretions of the oral cavity and the alimentary tract and gastric acidity prevent the bacterial colonization. Then comes the effective drainage, where the natural passages of the hollow organs like respiratory, GIT, urinary or genital system produce a way to drain the excretions effectively. Similarly, the ducts of various glands are the conducts of the drainage of the secretions. Obstruction in any of this causes infection. Then comes the immune defense mechanisms. They include the phagocytic leukocytes of the body, of the blood, phagocytes of the tissues and the immune system. This is a chart which shows the various methods of identification of the microorganisms. Bacteria, they are done by gram stain for bacteria, acid fast stain for mycobacteria, nocardia, GIMSA for the campylobacteria. The second entity, it is the fungi, silver stain for the most fungi, periodic acid shift for most fungi and mucicarmine for the cryptococci. Then comes the parasites, GIMSA for the malaria and leishmania, periodic acid shift for the amoeba and silver stain for the pneumocystis. Then all classes including the virus, culture, in situ hybridization, DNA analysis and polymerase chain reactions are done. Here in this first photograph we can see the gram stain which shows the bacterial colony in the gangrene foot. In the second photograph we can see the gel Nielsen stain for or AFB stain for tubercular bacilli. In the third photograph, it is the GIMSA stain which shows H. pylori. In the fourth photograph, we can see the periodic acid shift for the aspergillus in the lung abscess. In the fifth, we can see the mucicarmine stain which shows the cryptococci in CSF. In the sixth, we can see the mucormycosis in the lung abscess. Now, this is a small chart which shows that various diseases caused by bacteria, spirochetes and mycobacteria. Here, typhoid by Salmonella typhi, plague by Yersinia pestis, whooping cough or Bordetella pertussis, anthrax by Bacillus anthracis, chancroid by Haemophilus ducrei, Denovenosis or granuloma inguinal by Campylobacterium donovani, gonorrhea by Neisseria gonorrhoeae, cholera by Vibrio cholerae, shigellosis, brucellosis, diphtheria, lobar pneumonia. All these are very important diseases which you have to remember. Here this is another table which is showing the various diseases by bacteria, spirochetes and mycobacteria. Same. The diseases are including are bronchopneumonia, bacterial meningitis by Neisseria meningitis and all. Then other staphylococcal infections which are caused by Staphylococcus aureus, epidermis and saprophyticus, E. coli infection by Escherichia coli, clostridial gases by gas gangrene by Clostridium perfringens, tetanus by C. Clostridium tetani, botulism by botulinum, etc. Tuberculosis by Mycobacterium tuberculosis, leprosy by Mycobacterium leprae, syphilis by Treponema pallidum, etc. Now we shall talk about few diseases. The first is the plague. It has been the great killer since 14th century and it is known to have wiped out populations of cities. World over, about 1000 to 3000 cases are reported annually. It occurs regularly in Madagascar and Brazil. However, it's not a serious health problem in the modern Europe, Australia and US. In India, about 50-year Kizikkan period, an outbreak had occurred in Surat in Gujarat in 1994. Coming to etiopathogenesis, it is caused by Yersinia pestilentiae pestis, which is a small gram-negative coccobacillus that grow rapidly on the most culture media. Identification can be done by fluorescence antisera methods. It's a zoonotic disease and it is spread by rodents, primarily rats, both wild and domestic, and others being the squirrels and the rabbits. Humans are the incidental hosts other than the rodents. The infection in the humans are occurred by rat flea or by inhalation. Here the organism enters the bloodstream and they reach the draining lymph node, and rather than being phagocyted by the phagocytic cells, they proliferate rapidly, giving rise to tender lymphadenopathy. This occurs within 24 to 48 hours of the infection and it is accompanied by chills, fever, myalgia, nausea, vomiting and marked prostration. If untreated, death occurs from the disseminated intravascular coagulation within 1 to 2 days with development of widespread petechiae and ecchymosis leading to gangrene, and hence the name Black Death. Death also occurs from multi-organ failure due to toxemia. Virulence of the organism is attributed to the elaboration of plague toxins, that is pesticin and lipopolysaccharide endotoxin. These are some morphologic features. First is the like bubonic plague, then septicemia, bubonic plague, septicemia, and this is here typhoid. Inhalational things will be there, pneumonic areas will be affected, and typhoidal stuff will be manifested. Flea bite, this is the flea bite, and this here, this is the rat flea. This rat flea will cause the flea bite, and then bubonic plague, which is the most common, then or maybe by inhalation, which is called the pneumonic manifestation for provided by the typhoidal, and this will call septicemia. Bubonic plague, it is characterized by rapid appearance of tender, fluctuant and enlarged regional lymph nodes, several centimeters in diameter, and have discharging sinuses on the skin. Microscopically, the features are effaced architecture of the lymph nodes due to necrosis and in and around like infected nodes, multiple necrotizing granulomas, and mononuclear inflammatory response. Masses of proliferating bacilli in sinusoids or lymph nodes are seen. Cellulitis in the vicinity might be present. Coming to the septicemic plague, this is a form of progressive fulminant bacterial infection associated with profound septicemia in the absence of apparent regional lymphadenitis. Then comes the typhoidal plague. It is a form of plague that is unassociated with regional lymphadenopathy. Lesions are as follows: necrotic foci in visceral lymphoid tissue. Necrotic areas in the parenchymal visceral organs are also seen. GI manifestations with diarrhea and pain in abdomen may also be seen. Coming to the pneumonic plague, it is the most dreaded form, and it occurs by inhalation of the bacilli from airborne particles of the carcass of animals or from affected patient's cough. It is characterized by occurrence of bronchopneumonia with the following conspicuous microscopic features: necrosis of alveolar walls, intense hyperemia and hemorrhages, numerous bacilli in the alveolar lumina, characteristic mononuclear inflammatory response with very scanty neutrophils. Coming to prognosis, if plague is treated early, death rate is very low. However, if it is allowed to evolve into septicemic plague, death rate is very high, that is up to 40%. And pneumonic plague has the worst prognosis, and it is always fatal with 100% death rate. Coming to anthrax, it is a bacterial disease of the antiquity that spreads from the animals to human. It is widely prevalent in cattle and sheep, but human infection is rare. Then this has been used for bioterrorism, other microbial diseases in the CDC category A list in the group, which includes the botulism, pneumonic plague, smallpox, tularemia, and viral hemorrhagic fevers. In India, anthrax in animals is endemic in most states due to large unprotected and uncontrolled livestock production. Coming to etiopathogenesis, the causative organism is Bacillus anthracis, and it is a gram-positive aerobacillus, and it is a spore-forming bacillus, and spores so formed outside are quite resistant. It occurs as an exogenous infection by contact with soil or animal products contaminated with spores. Depending upon the portal of entry, 3 types of human anthrax is known to occur. Cutaneous form by direct contact with skin, and this is most common. Pulmonary form by inhalation, it is also known as Wolff-Otis disease and can be fatal. GIT form by the ingestion, and it is quite rare. The mechanism also includes there is a spread of the bacilli from the portal of entry to the regional lymph nodes through lymphatics, where the bacteria proliferate. There are delayed accumulation of polymorphs and macrophages. Macrophages also play a role in the expression of the bacterial toxicity. Bacterial toxicity is quite lethal to the macrophages. Coming to the morphologic features, the lesions can be hemorrhagic, edematous, or necrosis at the portal of entry. Coming to the first, that is the cutaneous anthrax, is the most common, and it is of 2 forms. One type is characterized by necrotic lesion due to the vascular thrombosis, hemorrhage, and acellular necrosis. The second type begins as a pimple at the point of entry of organism and into the abraded exposed skin, and most often in the regions of the hand and the neck Coming to the pulmonary anthrax, Wolff-Otis disease occurring from inhalation of spores of Bacillus anthracis in the infectious aerosols results in the rapid development of malignant pustule in the bronchus. This is followed by development of primary extensive necrotizing pneumonia and hemorrhagic mediastinitis, which is fatal. Coming to the third, that is the intestinal anthrax. It is quite rare and similar seen to cattle. Septicemia and death is most commonly seen. The lesions contain mucosal edema, small necrotic ulcers, massive fluid loss, and hemorrhagic mesenteric lymph node lymphadenitis. It also shows that spread of the infection to other organs also. Coming to the laboratory diagnosis, it can be diagnosed by few simple techniques. Smear examination. Gram stain smear shows rod-shaped spore-forming gram-positive bacilli. Endospores are detectable by the presence of unstained defects or holes within the cell. Culture. It grows on the sheep blood agar as flat colonies with irregular margin, medusa head, and anthrax contaminated work surface materials and equipment must be decontaminated with 5% hypochlorite or 5% phenol solution. Coming to the whooping cough or pertussis. Whooping cough is highly communicable disease and acute type of which is seen in the childhood. The use of DPT vaccine has reduced the prevalence of the whooping cough in the different populations. The causative organism is B. pertussis, Bordetella pertussis, which is a strong trophism for brush border of the bronchial epithelium. They stimulate the bronchial epithelium to produce abundant tenacious mucus. Within 7 to 10 days of the exposure, catheteral stage begins, which is the most infectious stage. There is low-grade fever, rhinorrhea, conjunctivitis, and excess tear production. Paroxysms of cough occur in the characteristic whoop. This condition is self-limiting and may cause death due to asphyxia in the infants. It produces a heat labile toxin, heat stable toxin, endotoxin, and a lymphocytosis producing factor called histamine sensitizing factor. Microscopically, the lesions are covered in the respiratory tract consist of a necrotic bronchial epithelium covered by thick mucopurulent exudate. Here, the peripheral blood smear shows marked lymphocytosis up to 90% and enlargement of the lymphoid follicles and in the bronchial mucosa and peribronchial lymph nodes. Coming to the next entity, that is the donovenosis. It is also known as granuloma inguinal or granuloma venerum, and it is sexually transmitted disease affecting the genitalia. And it is also in the perianal and inguinal regions caused by the Calmycobacterium donovanae. It is common in tropical, subtropical countries such as New Guinea, Southern Africa, Australia, and Asia. It usually inhibits the intestinal tract. Also, it is transmitted through vaginal or anal intercourse by the autoinoculation. Incubation period is 2 to 4 weeks. Here the lesion is in the form of papule, a subcutaneous nodule or an ulcer. Then it develops to a raised, soft, painless reddish ulcer with exuberant granulation tissue. Genitalia are involved in 90% of the cases and inguinal and anal region in 10%. Regional lymphadenopathy generally doesn't occur. Here these are the photographs. First is like the macroscopic gross ulcer which can be seen in the genitalia here. This is the bacilli. Coming microscopically, the margin of the ulcer shows epithelial hyperplasia. The ulcer bed shows neutrophilic abscesses. The dermis and the subcutaneous tissues are infiltrated by numerous histiocytes and containing many bacteria called Donovan bodies and lymphocytes, plasma cells, and neutrophils. These organisms are best demonstrated by silver impregnation techniques. Coming to the lymphogranuloma venerum, it is sexually transmitted diseases by Chlamydia trachomatis and it is characterized by mucocutaneous lesions and regional lymphadenopathy. It is no more considered as filtrable virus, it was previously thought, but it is instead given as intracellular gram-negative bacteria. It is worldwide in distribution, but prevalence is high in tropics and subtropics in Africa, Southeast Asia, and India. It begins as a painless herpes-like lesion on the cervix, vagina, or penis. The organisms are carried via lymphatics to the regional lymph nodes. The involved lymph nodes are tender, fluctuant, and may ulcerate and drain pus. Microscopically, the lymph nodes have a characteristic stellate-shaped abscess surrounded by a zone of epithelial cells. Healing stage of the acute lesion takes place by fibrosis and permanent destruction of the lymphoid structure. Coming to the cat scratch disease, it is another condition related to LGV or the cat scratch disease. It is caused by Bartonella Hensley, an organism linked to rickettsia, but unlike rickettsia, this organism can be grown in culture. It occurs mostly in the children under 18 years of age, and there is regional node enlargement which appears 2 weeks after cat scratch and sometimes after thorn injury. It is self-limited and regresses in 24 months. Microscopically, the changes in the lymph node are initially there is a formation of the non-cascading sarcoid-like granulomas. Subsequently, there is a neutrophilic abscess surrounded by palisaded histiocytes and fibroblasts. An appearing stimulating LGV discussed. The organism is extracellular and can be identified by the silver stains. Now, coming to one of the most important, that is the staphylococcal infections. Staphylococci are gram-positive cocci which are present everywhere in the skin, umbilicus, nasal vestibule, stool, etc. 3 species are pathognomic to the human beings, that is Staph aureus, Staph epidermidis, and Staph saprophyticus. The most staphylococcal infections are caused by Staph aureus. These infections are common in the commonest in the antibiotic resistant hospital acquired infection in surgical wounds. Caused by a wide variety of suppurative diseases are caused by Staph aureus. It includes meningitis, folliculitis, infection of the respiratory tract, bacterial endocarditis, septicemia, osteomyelitis, arthritis, and infective burn of the surgical unit. These are the suppurative diseases caused due to Staphylococcus aureus. Now coming to each one, that is infection of the skin. Staphylococcal infections are quite common. It begins from the dislodgement of the cocci from in the hair root due to poor hygiene and results in the obstruction of the sweat or sebaceous gland duct. This is known as folliculitis. If the follicles are involved and it results in a larger lesion, it is known as furuncle. If there is a spread of infection horizontally under the skin and subcutaneous tissue, it is known as carbuncle or cellulitis. Styes are staphylococcal infection of the sebaceous glands of Zeiss, the glands of the mole, and the eyelash follicles. Impetigo is another type of skin infection. Infections of the burns and the surgical wounds. These are quite common due to the contamination of the patient's own nasal secretions or from the hospital staff. Elderly, malnourished, obese patients and neonates have an increased susceptibility. Then comes the infections of the upper and the lower respiratory tract. This comes under the children under 2 years of age, and they get staphylococcal infections of the respiratory tract very commonly. They include the pharyngitis, bronchopneumonia, staphylococcal pneumonia in its complications. Then comes the bacterial arthritis in the elderly. Then the infection of the bone or the osteomyelitis in the young boys who have a history of trauma or infection. Then comes the bacterial endocarditis, which is acute and subacute bacterial endocarditis, which are the complications of the infection with Staph aureus. Then comes the bacterial meningitis. Surgical procedures or CNS may lead to the meningitis. Septicemia. It may occur in patients with lowered resistance or in patients having the underlying staphylococcal infections. They may present as features of bacteremia such as shaking, fever, and chills. Toxic shock syndrome, which is a very important topic, and it is a serious complication of the staphylococcal infection characterized by fever, hypotension, and exfoliative skin rash. This condition affected young menstruating women who are using tampons of some brands, and it kept in the vagina absorption of the staphylococcal toxins from the vagina. Then comes the streptococcal infections. It is also gram-positive cocci, but they are known for the non-suppurative autoimmune complications than the suppurative inflammatory responses. They occur throughout the world, but the problems are greater in underprivileged populations. The following groups and subtypes of the streptococci have been identified and implicated in various diseases. This photograph shows the various manifestations which can be seen as meningitis, pharyngitis, aphthous ulcers, pneumonia, lung abscess, subacute bacterial endocarditis, glomerulonephritis, septicemia, puerperal sepsis, and impetigo. Number 1, that is the group A or Staphylococcus pyogenes, also known as beta hemolytic streptococci, are involved in causing upper respiratory infection and cutaneous infections. In addition, beta hemolytic streptococci are involved in autoimmune reactions in the form of rheumatic heart disease. Group B or Streptococcus agalacti, it produces infections in the newborn and are involved in non-separated post-streptococcal complications such as rheumatic heart disease and acute glomerulonephritis. Then comes the third group C or G and G streptococci, which is responsible for the respiratory infections. Group D or Streptococcus faecalis, where it is also known as enterococci because of its causation of urinary tract infection, bacterial endocarditis, septicemia, etc. Untypable alpha hemolytic streptococci, which constitute the normal flora of the mouth and may cause bacterial endocarditis. Then the pneumococci or Streptococcus pneumoniae are etiologic agents for bacterial pneumonitis, meningitis, and septicemia. Now coming to gas gangrene, it is a rapidly progressive and fatal illness in which there is myonecrosis of previously healthy skeletal muscle due to the elaboration of the myotoxins in some species of Clostridia. The sources of myotoxin is Clostridium perfringens type A, others are Clostridium novae and septicum. Then comes tetanus or lockjaw, which is a severe acute neurologic syndrome caused by tetanus toxin, tetanospasmin, which is also a neurotoxic enterotoxin elaborated by Clostridium tetani. Then comes botulinism. It is characterized by symmetric paralysis of cranial nerves, limbs, and trunk. The condition occurs following the ingestion of the food contaminated with neurotoxin of Clostridium botulinum and are less often contaminated by the contamination of the penetrating wound. Clostridium food poisoning, it is caused by enterotoxin elaborated by Clostridium perfringens. And then comes the necrotizing enterocolitis or pig bell, which is caused by beta enterotoxin produced by Clostridium perfringens type C. Grossly, the disease affects small intestine segmentally. The affected segment of the bowel shows green necrotic pseudomembrane covering the necrotic mucosa and is associated with peritonitis. Advanced cases may show perforation of the bowel wall. Then comes microscopically, there is a transmural infiltration by acute inflammatory cell infiltrate with changes of mucosal infarction, edema, and hemorrhage. The pseudomembrane consists of necrotic epithelium with entangled bacilli. Then comes the diseases caused by fungi. Large number of human fungi are also known for infective as human beings. They are opportunistic mostly and they occur in conditions with impaired host immune mechanisms. Such conditions include defective neutrophil function, administration of corticoids, immunosuppressive therapy, and immunodeficiency states. This is the chart which shows the various diseases by fungi: mycetoma, aspergillosis, blastomycosis, candidiasis, coccidomycosis, cryptococcus, histoplasmosis, rhinosporidiosis, and superficial mycosis. Coming to mycetoma, it is a chronic suppurative infection involved by limb, shoulder, or any other tissues and is draining sinuses. The material discharged from the sinuses in the form of grains consisting of colonies of fungi or bacteria. Mycetomas are of two types. They are caused by actinomycosis and it is also known as actinomycetoma, comprising of 60% of cases. Eumycetoma is caused by two fungi, Madurella mycetomatis or Madurella grisi, comprising of remaining 40% of the cases. Eumycetomas are particularly commonly present in the northern and the tropical Africa, southern Asia and tropical America. Coming to the morphologic features, after several months of infection, the affected side, most commonly foot, is swollen and hence named Madura foot. The lesions extend deeply into the subcutaneous tissues along with fascia and invade the bones. They drain through sinus tracts which discharge the purulent material and black grains. The surrounding tissue shows granulomatous reaction. In this following photograph, we can appreciate the various abscesses, the brown granule and the necrotic tissues. This is a photomicrograph for the Madura foot. Then comes the candidiasis. It is an opportunistic fungal infection caused by Candida albicans and occasionally by Candida tropicalis. In human beings, Candida species are present as normal flora of the skin and mucocutaneous regions, intestines and vagina. The organism becomes pathognomic, which are balanced between the host and the organism, and it is disturbed. Few predisposing factors are impaired immunity, prolonged use of oral contraceptives, long-term antibiotic therapy, diabetes, obesity, pregnancy, etc. Coming to the morphologic features, it may present as oral thrush, which is the most common form of mucocutaneous candidiasis in the early life. Full-fledged lesions show creamy white pseudomembrane composed of fungi covering of the tongue, soft palate and buccal mucosa. Sometimes ulceration may be seen. Candidal vaginitis, it is a monolial vaginitis. It is characterized by clinically thick yellow curdy discharge. The lesions form pseudomembrane of fungi on the vaginal mucosa. They are quite pruritic and may extend to vulva and the perineum. Coming to cutaneous candidiasis, it's seen in the naine folds causing the change in the shape, paronychia, and colonization in the intergenous areas of the skin, groin, infra and intermammary intergluteal folds, and interdigital spaces caused by some forms of cutaneous lesions. Systemic candidiasis is rare and it is usually a terminal event of an underlying disorder associated with impaired immune system. They gain entry into the body through the ulcerative lesion of the skin and mucosa and may be introduced by a iatrogenic, which means intravenous infusion, peritoneal dialysis or urinary catheterization. The lesions of the systemic candidiasis may commonly encountered in kidney as an ascending pyelonephritis and the heart as candidal endocarditis. Here this is the photographs which shows the various inflammatory cells here. Here the spores are seen and this is the hyphae. These are the hyphae. It's a candidiasis of the ulcer in the skin. Coming to the cutaneous superficial mycosis, dermatophytes cause the superficial mycosis of the skin. And the examples are microsporum, trichophyton and epidermophyton. These superficial fungi are spread by direct contact or by formites and infect the tissues such as skin, hair and nails. Some of the examples are Tinea capitis, Tinea barbae and Tinea corporis. Tinea capitis, which is characterized by patchy alopecia affecting the scalp and the eyebrows. Tinea barbae by the acute folliculitis of the beard. Tinea corporis by the formation of erythematous papules. This diagnosis is mainly done by light microscopic examination of the skin scrapings after addition of the sodium or potassium hydroxide solution. Fungal culture and demonstration of the fungus may also be done in the tissue sections. Thank you for today. We shall continue the part 2 for the next class.